Hello, good afternoon, everyone. Today we are on the second part of the muscle physiology in our orientation class. So, we will start with types of muscle fibers. Now, these types of muscle fibers of a skeletal muscle depends on a number of factors. First is how does it look under a microscope and what are the structural components of it, and related to that what are the applied part of it, like where is it located and exactly for which function they are used. These things we are going to start today with. So types of muscle fiber, they are primarily of two type, type 1 and the type 2. The type 1 muscle fiber is also known as the slow type or the slow muscle fiber, whereas the type 2 is the fast type. Now, again, in this type 2, there are two subdivisions like type 2A and type 2B. Type 2A and type 2B. Like both are fast in type 2, but this division, the type 2A and type 2B, this actually has been classified depending on how fast they go into fatigue. Okay, now going by the characteristics. So we will write down type 1, type 2A and type 2B. So going by the metabolism, first is metabolism type. Now this type 1 muscle fibers, skeletal muscle fibers, they are oxidative in nature. Type 2A, it is like oxidative glycolytic, whereas type 2B, it is glycolytic. Now going by the type, like how fast a twitch is. Twitch, again we will come after later what a twitch means. So the type of the muscle, type of muscle. Usually this type 1, this is slow type, whereas the type 2, both of them, they are fast and fast. Okay, this is in terms of twitch, like how fast there will be a wave of contraction and a wave of relaxation. Now, the next is how fast this muscle will fatigue, like how fast there will be accumulation of the lactic acid. So, fatigue or fatigability. In type 1, this is like fatigable. It will fatigue, but it will take a long time to fatigue. Whereas this type 2A, it is fast and relatively fatigue resistant. Whereas this type 2B, it is again fast but fatigable. Now, coming to how does or what color do they usually look, even if we are seeing them with our naked eye. The color of this type 1 muscle fiber, it is red. It is like reddish, pinkish reddish in color. Type 2 is also red, whereas the type 2B, it is like very, very pale, rather whitish in nature. Now, if we remember the color, we can also correlate that this color is been because of presence of certain characteristics. Say for example, mitochondria and myoglobin. Okay, mitochondria and myoglobin, they are high in this red color muscle fibers. And also the capillaries. This capillaries usually which gives the pinkish tinge, the reddish tinge. Pinkish tinge. Chat is not actually visible to me. I can see the chat. Okay, now I saw. Yeah, hello everyone. Yeah, Anshul, thank you. Happy Teachers Day. Thank you. Thank you. Yes. Hemoglobin, what, G11 prediction? See, yeah, it's reddish because not of the hemoglobin. What does muscle have? What did we learn? Oh, G11 prediction. The thing is that, yeah, happy Teachers Day. Thank you. Thank you, Farhasik. Thank you for wishing. So the thing is that capillary is one factor. The capillaries are giving that pinkish color, and also the myoglobin, which is a pigment of the muscle. Okay, these are high in quantity, whereas in the type 2, it is moderate. Moderate means it is less than the type 1, but it is higher than the type 2b, and here it is lesser. It is least. It is present, but it is least compared to the other two. That's why the type 2b muscle, they look like whitish in color. Now coming to the metabolism. Metabolism is like glycolysis and oxidative phosphorylation, right? So first, this is very easy to remember. If we remember the name itself, right? The type 1 muscle, they are oxidative. So obviously here the oxidative phosphorylation rate will be high, and it will be like, intermediate, and it will be low. Okay. And coming to glycolysis, see, type 2, these are glycolytic, but in that type 2b is more glycolytic, right? So type 1, here it is very slow. Then this one, the type 2b is moderate, where type 2a is moderate, where type 2b, it is high. Okay, this is a way to remember. Now, the main function of the muscle fiber is what? To carry out contraction and relaxation, right? We have studied the sarcomere. Now, to carry out contraction or relaxation, what is mostly needed? What is mostly needed? The components or the activity that is mostly needed are of the calcium pumping capacity, because we know, we will study also right after this, calcium is essential, is absolutely essential for contraction of muscle. So calcium pumping capacity and myosin activity is very much important for the process of contraction and relaxation. So we know that this type 1 muscle fiber, that is slow type, means it is like the twitch is happening. Twitch means one contraction, one relaxation. That is happening, but that is happening slowly. That is happening slowly. So the force, the power, or the rate of the calcium pumping activity and the myosin activity, we can easily understand it is low. It is not absent, because without myosin activity or without calcium pumping activity, contraction can't happen, but it is low. Whereas in the type 2b, it is like high, high, and here also obviously it is high. Okay. Coming to the use part of it, why do we have to study and know what type of muscle fibers and what are they? Because depending on what type of activity I am doing, this muscle will be recruited. This muscle will be recruited. So, recruitment. Recruitment theory. Today, this one we will understand. So, just now, for now, you just remember the meaning of recruitment means whenever there is any stimulation from the nerve that comes to the muscle, because each and every muscle is innervated by a nerve fiber. So, when the stimulation comes from the nerve to the muscle, which muscle gets the wave of contraction faster? Gets the wave of contraction faster. That is called recruitment. It is getting recruited. It is getting recruited. Okay. Now, here, this slow type of muscle, it gets recruited first. Okay. Then, type 2b, it gets recruited next, and the last to get recruited is the type 2b. So type 1, type 2a, type 2b. Okay. Now, in what activity do we use this muscles for? Like suppose I'm sitting, I'm standing, certain type of skeletal muscle will be used. If I start running, then a different group of muscle fiber will be activated. If I'm an athlete and I'm sprinting, doing the sprints, then also the muscle fiber that will be undergoing contraction to provide me the energy to perform that activity will be different. Okay, so we write it as type of activity involved in. So the slow type, the type 1 is a slow type. Suppose I'm just sitting idly or standing idly, then this is the muscle which will undergo contraction and give me the mechanical strength and enable my muscles to allow me to stand. So, this is standing. The activity in which this type 1 slow type muscle is undergoing contraction, is participating, is standing. Okay, standing, sitting, all this static position where I'm not moving. Okay. The type 2A, when I am running, suppose all of a sudden I start to run, then there is contraction and relaxation of the bicep, tricep and the calf muscle, gastrocnemius muscle, all these muscles are involved. So, there the type of muscle that will enable my limbs to do the act of running is the type 2A muscle fiber. Okay. And type 2B, this is for high exercise, like heavy exercise, like sprinting, like sprinting. Okay, these are the type of activity they are involved in. So, depending on the characteristic, these are the classification of the muscle fiber. Now, we come to the very interesting process, that is the excitation contraction coupling. Okay. Yesterday, we have understood the neuromuscular junction. We have also understood that for the process of contraction, the first start point is when an impulse will flow all the way to the axon and release a neurotransmitter, excitatory neurotransmitter, and it will relay the wave of depolarization to the to the post-synaptic membrane, that is the sarcolemma of the muscle fiber, right? So, a very quick revision of that, like just let us mention about that, and then we go to the process of excitation contraction, because when we study the excitation contraction process, we would be separately understanding what happens to the actin molecule, then what happens to the myosin molecule, and then we will club these two processes together and understand the whole excitation contraction process as a one. So before we start that, just a quick run through of the yesterday's process very quickly. So what was there? First, what happens? So first, what happens? First, there is action. No, Varun Kashyap, no, my screen is not hiding. Others, can't you see my screen? Varun Kashyap, I'm talking, means other students were like Anshul, Farha, G11 prediction. You guys can't see the screen? I'm not hiding the screen. If anyone else can't see the screen, just drop a text. So action. No, okay, Ash, you can see, right? Okay, fine. Then Varun, I think you have to refresh your page or something. So action potential. I'm just writing briefly because this total detail we have studied through the presynaptic membrane. Action potential through the presynaptic membrane. What it does, it causes release of acetyl-CoA. Now, this acetyl-CoA, we know it's an excitatory neurotransmitter. I'm not going into that. Release of acetyl-CoA where in synaptic cleft by the process of exocytosis. Exocytosis. Okay, the ionic basis we know. Okay. Now, once it is done, what will happen? End plate. Okay, Anshul, visible? Yeah, thank you. Chirag, yeah, it's visible. Thank you. End plate potential generated. So once end plate potential is generated, then what is the big step? Action potential swaps, means goes to the post synaptic membrane. Now here the postsynaptic membrane is obviously the sarcolemma, the muscle, the skeletal muscle region. So once that is over, what will happen? Sodium channel, Hodgkin cycle that we studied in homeostasis. Sodium channels will open. Sodium channel opened, influx of the calcium first through the voltage gated, then through the non-voltage gated, that is dihydropyridine receptor as well as ryanodine receptor. So all together, this influx, where will it come? Calcium will be pulled out of terminal cisternae into the sarcoplasm. Okay. Now, this is like the main trigger. That is the main trigger for our actual preparation for the contraction to happen. So once calcium is pulled off to the sarcoplasm, now calcium, what do we know? When calcium is present anywhere, it will not sit quietly. It will always do something or the other. Now step by step things will happen simultaneously. So first we will see activation of the myosin molecule. Which myosin molecule? The subtype 2, because in skeletal muscle this is what is present. Okay. Now, if we draw it briefly for our understanding, say this is the tail of the, I'm just drawing opposite. Okay. Tail. Now, say this is the neck region and this is the myosin head. right? Now, in the myosin head, what is attached during rest? That is during resting condition when there is no contraction. It has a ATP attached to the myosin head. This is the head, this is the neck, and this is the that box thing. It is the tail. That is the heavy chain. Okay. How is it lying? We from the sarcomere thing we already have an idea. So it is like lying in context to the actin molecule, but there is no physical contact with the actin, right? There is no physical contact with the actin. Myosin is lying by itself, actin is lying by itself. Now, when there is that time for myosin to get activated, what will happen? This ATP that is bound to the myosin molecule, it will undergo hydrolysis of ATP by the ATP phase. Because we have studied myosin head, it has got a catalytic site and a regulatory site. So that is the catalytic site in which the ATP is bound. So once the ATP is hydrolyzed by ATPase, then thing that will happen is this myosin head. Now here it will be ADP and IP because ATP is broken. Now this myosin head, which is now having ADP and IP after ATP has broken down, it is called the powered myosin. Now, the myosin is activated. It has gained powered. But just at this step also, there is actually no binding with the actin. Okay, now this myosin is ready to bind onto the actin. Now, when this is happening, the lifting of the actin active site is also done. Now, activation of the binding site in actin. So, at rest, okay, in resting condition, how does the actin lie? We have seen in resting condition, the actin lies like this, in which this globular structure, this is the F actin double helical structure, the polymerized G actin into F actin, which is double helical in structure, and this one is tropomyosin, and there is troponin. There is a troponin molecule. There are this troponin molecule which is covering the active site of the actin. So, if just for our understanding purpose, if suppose I highlight this is one actin molecule and this is our troponin molecule. So, this troponin is covering the binding site on the actin. Okay, so this is the site on the actin on which the myosin is supposed to bind. So, this is the actin and this is the troponin. Now, here the subunit with which this actin is covered, the binding site on actin is covered, it is the I subunit of troponin, right? Other than that, there are two other subunit on troponin. Like this is one subunit and there is another subunit. Now this is the T subunit and this is the C subunit. And this was a subunit through which it is covering the active site on the actin. Now, all this site, they are actually in physical contact with each other. They are in like very much physical contact with each other. They are touching each other. So, upon release of calcium into the sarcoplasmic reticulum, upon calcium release, what will happen? We know the C subunit of the troponin. This is for the calcium to bind, right? So now what will happen is, again let us draw. This is our actin and say this is our troponin and the subunit C now binds with the calcium. And when it binds with the calcium, what it does is that the other subunit which was covering the active site on the actin by the I subunit, it is like pulled away. It is like pulled away. After pulling it away, it exposes the active site. Like after pulling it, what it does, it not only pulls, it causes like swaying of this inhibitory site like upwards. It causes like swaying of this inhibitory site upwards. Calcium is bound. So, unless and until the calcium is not bound to the troponin C, this exposure of the active site will not happen. Okay. So, calcium has come to the sarcoplasm. The binding site on actin molecule on which the myosin will bind is exposed, and while this was happening, in the meantime we saw how the myosin also got empowered. Means basically hydrolysis of ATP has happened and it has broken down into ADP and IP. Now both the molecules are ready for binding onto each other. Means the myosin will go and bind onto the active site of the actin and pull it, right? That's what we studied from the sarcomere. Now, how does the EC coupling happen? Both are now empowered. Now, this is the actin molecule. First, I will draw and show for your understanding. Then I will show you the picturesque diagram from the images. That will lay a very good concept for you guys. Okay. So, now the empowered myosin, the actin. So, this is the binding site. So, this is the binding site on the actin on which the myosin has to bind, and this is our empowered myosin. So, now first what will happen? This myosin, it will go and bind on the binding site on actin. We already know, still I am writing for our concept clear. So, now here you see this actin and myosin, the configuration at which they are lying, that is at a degree of 90 degree. It is at a 90 of 90 at a degree of 90 degree. At a degree of 90 degree, the myosin head goes and binds on the exposed site on the actin. Now this configuration is also called as the cross bridge configuration. Okay. After this binding happens, then the actin, it stays as it is. The actin does not move. So this actin is something like what we have seen here also. It is lying as it is. Sushant, good afternoon. You are late by few minutes though. So now what happens? This after this myosin molecule binds on the actin at 90 degree. After that, so this is the actin on which it is binding. After that, the shape, the position of the myosin is changed by the neck. The neck portion is the one which is flexible, which is mobile of the myosin. So now this myosin head, it assumes an angle of 45 degree. It assumes an angle of 45 degree and gives a power stroke. Now the energy condition, the energy configuration, there is an energy thing in it. The ADP and the IP that is attached play a very, very important role. So when the myosin head, it just touched the actin, that time the myosin molecule, it had ADP and IP attached to it when it just touched at the angle of 90 degrees. Now, when the neck was bent and a 45-degree configuration, like the actin is lying as it is, but the myosin is now bent its neck and at an angle of 45 degree, it is touching the actin. This is the stage when the ADP and IP, they dissociate, they get detached, they get detached. So here detachment of ADP and IP from myosin head happens. Okay, now this is what we call as the power stroke. So this is the power stroke. That is that pushing of the myosin head at an angle of 45 degree on the actin. And this power stroke, this is very, very essential to remember. Power stroke will only happen when the ADP and IP are detached, Now, this complex, let me highlight it. Okay. Okay. Now this complex means, see, the structure in which the actin and the myosin, they are a complex. They are not physically in contact with each other, right? Through the binding site. Now this complex is called as this complex is called as the actomyosin complex. Okay. Here, the actomyosin complex is very rigid. Very rigid. The myosin head without ATP, when it is bound to the actin, it is so powerful. That complex is so strong that it cannot get dissociated by anything or on its own. It's a very, very strong complex, and that is what causes the power, causes the contraction. Now, outcome of it, what happens due to this power stroke? Due to this power stroke, 11 nanometer of actin is pulled towards the myosin. Okay, like that it pulls. That's why You remember we studied in the sarcomere, what did we study? That when there is contraction, during contraction, again a quick reminder, what happens? The actin molecules, they get pulled towards the myosin, right? So this is what happens. The myosin, after the power stroke, it pulls the actin towards itself by 11 nanometer, and that's where the during contraction the length of the muscle also reduces. And see here, it is an energy consuming process, right? Energy consuming process. So ATP is consumed. Another point to remember, whole of the ATP is not used. Okay. So what happens to the remaining? The fraction of ATP that is unused dissipates as heat. So whenever the muscle is contracting, the muscle is relaxing, the muscle is not doing anything, there is always little bit of heat generated, because this is the reason. For every activity of the muscle we need ATP, but this ATP is not totally used up. Okay, so this is our EC coupling. Now there is a this terminology. I'm coming to the relaxation part, but see the way they are like lying, this myosin and actin, right? So here actually during contraction, the actin slides over myosin, right? The actin is like sliding over the myosin. Since this happens, thus it is also called as or defined as sliding theory. Sliding theory of the muscle. This is what happens during contraction. Okay, so last what did we study? That the actin and the myosin, they are forming the actomyosin complex, which is like very, very rigid, and there is nothing that can unbound them. So now how does relaxation happens? So relaxation, that is the unbinding of actin and myosin. For the contraction to happen, we needed one myosin. Why? To empower the myosin, to make the myosin powerful, we needed one breaking down of ATP, right? One breaking down of ATP. Now, to unbind, to break the actomyosin complex, we need one ATP, but that ATP we don't need to break down. How do we need? Like this actin molecule is lying like this, right? Lying like this. And the acto molecule complex is what? If this is the binding site and this is like, this is how the myosin is, right? The actomyosin complex. Okay. Now, one ATP molecule, one ATP molecule will go and bind on the myosin head. So, as soon as one ATP molecule is binding on the myosin head, it will just again make the myosin head powerless. It will make the myosin head powerless. So immediately the myosin head now onto which the ATP is bound, the myosin head onto which the ATP is bound, immediately it will be pulled back away from the actin. It will be pulled back away from the actin, thus relaxation. Now, relaxation is the unbinding of actin and myosin. True, very true. But we also have to remember the agent or the substance which is absolute necessary for excitation and contraction is what? Calcium. So, calcium is absolute necessary. Now, it is just before the excitation contraction that calcium has come into the sarcoplasm. Sarcoplasm means muscle fiber cytoplasm, right? So, after the calcium has caused the excitation, contraction, it is not that all the calcium has been used up. No, it doesn't happen that way, right? And it's an ion. So, when the relaxation have to happen, the calcium have to also go backward. Backward means it has to also withdraw the steps that it has done. Like on the part of the actin molecule. So this is the actin molecule. Again, the troponin that was bound to it, like with the C, with the T, and with the I, right? Now, calcium will get dissociated, will move away from the C subunit of the troponin. So, once calcium dissociates from the C subunit of troponin, right? The calcium dissociates from the C subunit of troponin. So when this happens, once again, this actin molecule, say this was the active site. Now again that active site will be covered by the, let's draw like that, it will be easy to understand, with the I unit, C and T. So again it will go back to its original form. So the actin site is closed. The myosin has also been powerless by binding of ATP. Now calcium has bound to the C subunit of the troponin, and once again the active site on actin is covered. Okay, this is what has happened to the actin and myosin. But there is lot of calcium that is still present in the sarcoplasmic reticulum. There are lot of calcium which is still present in the sarcoplasmic reticulum. And if calcium is present in the sarcoplasmic reticulum, there are always very high chances that there will be again trigger for this excitation and contraction. So now the pumps, role of pumps in relaxation. Okay, what will the pump do? We have studied that we have a pump in last class, yesterday's class, sarca protein. Now, the sarca protein which is present, it will pump the calcium from the sarcoplasm into the sarcoplasm reticulum. Okay, now sarcoplasmic reticulum is the storehouse, right? So it will pump it back to the sarcoplasmic reticulum. This will do. Again, there is another pump, another buffering pump. What is that? That is calsequestrin. The calsequestrin, it will not remove the calcium back into the sarcoplasmic reticulum, because sarcoplasmic reticulum is the storehouse of the calcium, right? So this calsequestrin, what it will do? It will buffer the excess calcium present in the sarcoplasm. So this is how the excitation and contraction following the sliding filament theory will happen. Like we have understood how the actin gets activated, we have understood how the myosin gets empowered, then we have seen how the binding of this myosin on the actin head happens. Then we have unfolded the relaxation also, unbinding, then inactivation of the actin, and additionally the pump that will remove the excess calcium from the sarcoplasm. Okay, hope this concept is clear. Any doubt? If you have any doubt, you can ask. Okay, this is the excitation contraction coupling. Now there is an applied part to it. What is the applied part? What is the applied part? Now, the applied part is what we call as rigor mortis. Rigor mortis, what is the definition of rigor mortis? It is the stiffening of the body muscles, skeletal muscles, obviously, post death. Okay. Now, this rigor mortis, it sets in 12 to 19, like till, it remains maximum, usually till 12 to 17. It may last to 12 to 19 hours also. It starts and lasts for 12 to 19 hours after death. What is it actually? How does it happen? In what sequence does it happen? Like which part of the body after death gets stiffened most? Now, as per Nisten's rule, cadaveric because it is studied in the cadavers. Cadaveric rigidity, which also means the rigor mortis, happens like starts with the mastication muscle, like the muscles of the jaw, the cheeks, and the chewing muscle. When we masticate our food, the muscles of the face, facial muscle that are used for chewing of food, mastication of food, it starts with that. That is the one that becomes hard first. Start with the mastication muscles. Then neck, then trunk and upper limbs, and lastly the lower limbs. Okay, this is how the stiffening happens. But the stiffening or the rigor mortis, it does not last forever and ever. After 17 to 19 hours, the body that's stiffening also starts becoming loose. It becomes like loosening up. So it is said that the passing of rigor mortis happens same as the setting of it. Okay. Now, what is the cause of the rigor mortis? Why all of a sudden after death for 12 to 19 hours or 17 hours, there will be this tightening or like, rigidity, cadaveric rigidity. What is the reason behind it? What causes it? Cause for rigor mortis. What is the cause of it? It is again the actomyosin complex. Why it happens? Some muscles at the time of death. So, as long as the person was alive, was breathing, some metabolism or the other was going on. So, even after that, there is little bit of cellular metabolism that goes off for some time. It does not stops abruptly, right? The body temperature or the metabolism, it gradually passes away. It stays intact for little bit of time. So, some muscles at the time of death are in actomyosin complex, right? The the actin and myosin are bound to each other at 45 degrees angle, and there is a power stroke. Now, this does not get dissociated. So, metabolism stops after death. So if metabolism stops after death, all the cellular respiratory chain, the oxidative chain, all the anaerobic and aerobic respirations, everything changes, stops. So when there is like stoppage of all this cellular respiratory activities, all the metabolites which gradually stops, then there is no ATP generated, no ATP generated, because all ATP gradually becomes used up. So, so there is like no ATP to bind with the myosin and deactivated from the actomyosin complex. So, once the ATP is not available, still there are certain skeletal muscles where the actin and myosin are bound. They are still bound. So those myosin which is still bound to the actin, they don't get ATP, by which the myosin head will be deactivated. So since there are no ATP for the myosin head to get deactivated, the myosin head do not come out. It is not pulled away from the actin. So in this condition, the actin and the myosin stay tightly as actomyosin complex, causing the rigidity, because the rule is that the high yield is that no ATP, no breakdown of the actomyosin complex. So until several hours they stay tight. Now what causes the passing away? Now, over a period of time, this muscle fibers, because they are also not getting any nutrition, no circulation, no oxygen, nothing. So they also start becoming loose. They also start to die off. This actin and myosins are what? They are proteins, right? So this protein, their activity due to stoppage of circulation, due to stoppage of the metabolism after death, several hours after death, they start to wear off. So that again causes the flaccidity, the loosening up, and the actin and myosin are unbound. Okay, this is the applied portion from the actin and myosin excitation contraction coupling. Hope it is clear to you all. Moving on to the, any doubt you can ask me, is another interesting concept, which is the motor unit. Okay, motor. Now, what is a motor unit? It's a unit. So, it means it has got more than one component. So, a a typical motor unit includes a ventral horn cell. It includes an alpha motor nerve. Now, from the term motor unit, we can understand it. It means that this unit is concerned with the motor function, the mobility function, not the sensory function. Sensory function means the one that we studied in the Erlanger Graces and then in the Lloyd Hunt also. Like, it is concerned with the feeling of senses like touch, pain, temperature, pressure, all those. But motor unit means the components in this unit, they are concerned with the motor function, the mobility, the movement. Afnan, can anyone tell this note? We to make new book or we can write it. You should do one thing, Afnan. You should just sit with it because this is a live session, a class. So you can sit with a notebook and you write down. As you are listening, you write it down. And then you can, as a reference, you can go and study any book. So if we are to draw a motor unit, it will be like this. So this one and okay. Now this is a ventral horn cell, that is the dorsal, this is a ventral. So there are, very simply, just for our understanding benefit, very simply a simple diagram. So these are the spinal alpha motor neurons, okay, which are going and they are innervating like muscle fiber. They are innervating various muscle fibers. Okay, this is what and how they look like. Okay. Now, this one, this one alpha motor nerve, it may innervate like like few, few muscle fibers. Okay. Or this single motor unit, that it can innervate many muscle fiber. Okay, so the whole bunch, like every time the spinal motor nerve plus the number of muscle fibers, it innervates, makes a motor unit. Okay. So each nerve fiber, be clear in it. Each nerve fiber that it is innervating each and every muscle fiber, that is one motor unit. Now here structures that have smaller motor unit, like the nerve fiber innervating lesser number of muscle fiber. The nerve fiber innervating lesser number of muscle fiber are ocular region, are in the ocular region and fingers. So basically structures that are involved with fine work. Okay. This is like fine thing. With fingers, what do? We write, we stitch, we draw. The fine, we sew, right? We knit. So, knit wool. So, the fine work that we do with the structure and ocular also, it is like very precise. It's for precise movement. It's not for lifting anything, right? It's only to see vision and move it around for the purpose of vision. So, this is a very precise activity. So, the number of nerve fiber innervating the structures for precise activity, they are less. Whereas the motor unit for the motor unit for large activity has more muscle fibers. Okay, like for gross activity, like the muscle of the back, muscle of the trunk, they have got like one motor fiber, one motor nerve fiber. They innervate like many skeletal muscle fiber, like one in this region, one motor fiber innervates about 500 to 600 skeletal muscle fiber. Okay. This is the motor unit. Now, what are the characteristics of a motor unit? What are the characteristics of a motor unit? Characteristics is that it follows all or none response. Like upon stimulus, either the whole motor unit will show action potential, undergo contraction, or none of it will show. It will not be that only few fibers from the motor unit are showing in normal healthy physiological condition and others are not showing. This will not happen. Okay, it will be a all or none response. Second important property is that it follows the size principle. Now, what is size principle? Size principle is Honeyman's principle. Okay. What does this Honeyman principle talk about? It says that, what does it say? It says that upon, what does it say? Upon stimulus, the small fibers, or rather small motor unit gets recruited first. The small motor unit gets recruited first. Okay. But upon withdrawal withdrawal of stimulus, the small motor unit gets derecruited first. Means when there is contraction stimulus, it is the smaller units like the motor fiber that is innervating only less number of fibers, they will get recruited. They will show the contraction first. But relaxation will be opposite. It is the relaxation will be, it is the larger motor fiber like which are having like more number of skeletal muscle fiber, they will get de-recruited first. This is what is the size principle. Now another thing that is important in context to the motor fiber. Now I have mentioned about twitch. Now what is a typical twitch? So if I am drawing a twitch, it will be like upon, so this is a stimulus of threshold intensity. So upon stimulus there would be like latent period, and then there would be a sharp rise followed by a quick fall. So the upward stroke rather, it should be little broader because the relaxation is for longer period. Okay, so a sharp rise followed by a quick fall. So this is the phase of contraction, and this is the phase of relaxation. Okay. Now, if more than one stimulus is given, then there may be summation. We know the criteria. If the second stimulus is in the relative refractory period, not absolute refractory period. If it is in the relative refractory period, then there can be summation like first one wave and then second wave. Right? If repeated stimulus is given, then what may happen? Then the muscle fiber may just go into tetanus, like constant repeated stimulus is given, it may just go into tetanus, and for a prolonged period of time there may be contraction until it falls down. Okay, so these are the some characteristics: summation and tetanus, like prolonged contraction which is not desired. Another thing is that when stimulus is increased in intensity, then gradually more motor units are recruited. Meaning what? Suppose first there has been one stimulus. So in response to this stimulus, say for example, there is one contraction of certain amplitude. Then again there is another stimulus of bigger intensity next. So there will be recruitment of more motor unit. Okay. Then again, there suppose the intensity is increased further. Then again, there will be another more recruitment, and again there will be increase in the motor unit recruitment. The size will go bigger. So like that, with gradual increase in the stimulus, bigger and bigger motor units get recruited. Okay, this is our concept of the motor unit. Okay, so Afnan, I was asking I should write this in a new book, or that's up to you. The main thing is for you to understand and remember. So whichever book you write, it's fine. So this is all for today. Anyone has any doubt? Tomorrow we would start our orientation class for the cardiovascular system heart. Okay. So anyone has any doubt in today's topic, you can write to me. Anything that you didn't understand? Okay. So I guess everything is clear. So then I will all see you tomorrow, same time with the orientation class for the cardiovascular system. Okay. Bye and have a, which one? Can you explain this again? Alphase, which one you mean? This means what? The recruitment. The recruitment means, like, suppose when there is stimulus, when there is a stimulus, a certain number of motor unit, like the smaller motor unit, like the nerve fiber which is innervating, less number of muscle fibers, they get recruited. Upon increasing the stimulus, more muscle fiber get recruited and they show more contraction. So if more number of muscle fiber are getting recruited, means are getting contracted, automatically there will be increase in the twitch. There will be increase in the force of contraction and the time of contraction also, the amplitude of contraction. Upon increasing the stimulus, then what will happen? Even more motor units will start getting contracted. So here again, the force of contraction, the twitch will be of bigger amplitude. The force of contraction will be more and stronger. So as we increase the stimulus, the motor units, because there are so many motor units, right? Every nerve fiber that is innervating the muscle fiber is a motor unit. So upon increasing the stimulus, more and more motor units starts getting recruited. Understood? Alphas, is it clear? So this is what it means. Upon increasing the stimulus, more and more force of contraction increases. So I will see you guys tomorrow again with orientation for the cardiovascular system. Okay. Bye and have a nice day.